everyone. Welcome back to our next session at the Longevity Leaders Conference. We've got a real treat for you today. Very important discussion looking at the roadmap to commercialization, particularly examining some of the clinical and regulatory pathways for longevity therapeutics. I'm very, very excited to hand you over to your panel. Before I do, just a reminder, please do put your questions and comments into the live chat so that the panel can address them as we go. We've got some real expertise in this room and looking forward to hearing what they have to say. But without further ado, I'm going to hand you over to your moderator for today's conversation, Alkahest Stephen Braithwaite. Stephen, over to you.
Thank you very much, Angela, and welcome everybody to this panel. It's a real pleasure to be moderating this discussion today. I sort of reflect back when I really started in the aging science field 6 or 7 years ago now. This was still a little bit of the fringe science, and there were still some concepts that were quite soft, and the development of the field over a relatively short time has been incredibly impressive, because there's very strong science now that is forming foundations of this field, and this sort of short period is now leading us to a time that we have to think all of those steps ahead to how are we going to translate the science to real therapeutics that make a difference to patients, and you've got to think those 5 or 10 steps ahead to commercialization, to the potential paths on the way, and it's a really fun phase to think how you navigate through to this endpoint, and so this is why this panel's a real pleasure to be moderating today as well. That we're having that sort of free-flowing thinking about how we get to the endpoint that we really want to, and here the panel is a group of people who are really involved in the developing therapeutics, actually in that phase of actually taking drugs and various therapeutics forward to engage in this discussion. Where are minds right now? Where are the things that we need to think about? So we really look forward to an open discussion from all the minds here. So a brief introduction about myself. I'm Stephen Braithwaite. I'm Chief Scientific Officer of Alkahest. We're a San Francisco-based company built on understandings of plasma as a therapeutic and a source of therapeutics for age-related disorders, with the hope in the long term of developing therapeutics which are really generalizable for a whole host of aging-related conditions. We have moved into phase 2 trials in Alzheimer's disease, Parkinson's disease, age-related macular degeneration, and other disorders with plasma fractions and small molecules. So we're really moving into this area very, very rapidly. And to me, the goal of longevity therapeutics are to really change the way we think about healthcare, to think in this more generalizable way that we can affect fundamental mechanisms of aging and then have a broad effect that goes beyond thinking one indication at a time into something much broader and generalizable. So the opportunities are huge eventually. We've got to navigate through that path. So with my background, I'd like to pass on and go further east with the rest of the team on the panel here, from myself in the San Francisco area. So I'd like to pass to Tom, if you could give a quick introduction to yourself and what you think longevity therapeutics are. What's your definition?
Sure, no, I'm happy to. And thank you, Stephen. Thank you also to the organizers for asking me to join here today. It's a very exciting question, I guess, to discuss. So yeah, my role is I'm CEO of a company called Navitor Pharmaceuticals. We're focused on developing a sort of new generation of compounds that impact mTORC1, and currently our sort of most, I'd say, age-related program that we're working on is a selective inhibitor of mTORC1 that the company has discovered, recently patented, and this is a fully selective inhibitor of mTORC1, and we're developing it initially for rare genetic kidney diseases as a sort of a way of advancing the programs forward that is sort of, let's say, attractive to investors in particular. Our ultimate goal, of course, is to explore the impact of these compounds more broadly. And we think there's tremendous promise for the class generally. We know that rapamycin itself is considered to be probably one of the most well-validated, well-established compounds that improves lifespan in animals. And so there's some adjustments in the sort of the specificity profile and other considerations around these compounds that might be of interest, and that's basically what we're working on. So when I think about longevity therapeutics, it's really, I think from my perspective, looking at that sort of constellation of chronic diseases that converge on people as they get older. And so it's the comorbid conditions, think about arthritis, think about diabetes, think about Alzheimer's disease, think about all of the inflammatory and fibrotic conditions, decline of systems generally that contribute broadly to healthcare costs tremendously, but also are real dampers on quality of life. And so for me, it's really asking the question of whether we can build better years toward the back end of our lifespan, where people can have more involvement, extended in interaction with their colleagues in their careers, but also better years, quality years with their families as they get older. So that's, for me, the mission.
Thank you very much, Tom. And pass over to Joan in Boston as well.
Sure. So Joan Mannick, Head of Research and Development at Life Biosciences, which is a company that has 3 platforms targeting different aspects of aging biology. I think when I think about longevity therapeutics, the bottom line is aging is just a biology and it's modifiable. And this is important because aging is the biggest risk factor for almost every chronic disease. And obviously, aging also determines our longevity. Because it's a modifiable biology that can be targeted therapeutically, this opens up a whole new areas of medicine. And these areas of medicine have the potential to treat or prevent or even reverse multiple different aging-related diseases and open up whole new areas of therapeutics, and that's why it's exciting.
Thanks. Thanks so much, Joan. David, I'm sure you're excited too. So please introduce yourself.
So I'm David Roblin. I'm a clinician scientist, and I work for Juvenescence. I'm the Chief Operating Officer, but the Chief Executive Officer of the Therapeutics Division. Juvenescence has got some broad interest. We've got commercial interest. We've just recently launched a ketone ester in our nutritional supplement business, Juve Life. And we've got a cell and cell therapy division and a prescription drug division, which is in the clinic and moving towards the clinic. And we also got a data sciences division, which is helping us decode very complex biology. It's a pleasure to be here. Thanks, Stephen and the organizers for inviting me. I'm not going to be terribly controversial in terms of longevity therapeutics, which is a shame probably for a round table, but in the first instance, for me too, it's about, I guess I saw someone write that it's about changing our, not necessarily changing our expiry dates, but the time leading up to it. So it is also, for me, about thinking about health span. And that is important for the reasons that have been described already. Life expectancy now is mostly determined by what happens in the elderly in the Western world in particular. And I think, some simple stats, those over the age of 65, around 50% of more than 2 chronic diseases. And of course, chronic disease predicts another chronic disease and predicts death. That's because I think fundamentally many of these diseases do share common signaling pathways. And we've come to look at some of them in this emerging science that we might describe as underwriting it all. So it's about health span. I do worry that as we treat disease, we might increase lifespan, but also increase disease span. So I think it's really important that we think about health span and we address the fundamental causes of aging. So when I sit there, I think a great goal would be to reach 100 in very good health. And I think that is something that we can aspire to. And our role in the companies that we represent here, I think, is to guide patients how to do this. I think we need to intervene if needed. We'll have to intervene early, likely in pre-disease. We're seeing some of that play out in the Alzheimer's disease area now. So, maybe we can talk about looking at prodromal elements. And most of all, actually, the other thing I'd like to say is if we do develop longevity therapeutics, when we do develop them, we can make them accessible to all, widely accessible therapeutics. So that's kind of the philosophy I bring into this. Great.
Thanks so much to the whole team there. I think that the health span concept is one that resonates with all of us, and the concern about just affecting one disorder and then you just get hit with the next one, and the next one is one that is a worry for healthcare of age-related disorders. But it is a reality that getting on this path to increase health span, we probably have to think one disorder at a time, and this is sort of the reality of pharmaceutical development to go to those endpoints of individual disorders, and then towards the generalizability as a later step. So maybe back to Tom again. How do you think of the going through individual age-related disorders and going for one disorder at a time first? How do you think about picking that disorder, or do you sort of think more generalizable early on?
Yeah, I mean, this is a really challenging matter, and I think probably, the way, at least the way that I would look at it is, if you have a therapy that has a particularly direct use in one indication, one should focus on that. And so, I'll just again use the example of our program, which we have a mTORC1 inhibitor that has a very interesting distribution within the body. It goes to the kidney very well. It goes to the liver. Also, the cardiovascular system is where we see the greatest amount of target engagement. And so, if you think about it, the patient population we're looking at, we'll be certainly focusing our development on kidney-related endpoints, but these patients also experience dysfunction, deficit in function of the liver, and they're known to develop a rather fulminant cardiovascular disease and heart enlargement and other issues. And so I think the way that I would see any of these drugs really being developed is you pick one that is relevant and where you can very confidently have a path toward registration. And at the same time, you collect stamps around that would inform you about markers of impact on these other disease states. And, they can either become profiling features, or they can become sort of label extensions, or however you might look at it. But it would be first to build the argument that there is a benefit, and then you could run the studies that would be properly powered to be able to demonstrate benefit there. I think this is, for example, the approach that, as I understand it, at least what BioAge is doing with some of their programs, is to have a path to approval for their molecules that they then will use that patient population to collect the data that would argue for broader benefits. I think it's a very sensible way of working at it. And that way you're not risking your entire development program on an outcome which is really unprecedented, which would be to have a treatment approved for multiple comorbid conditions where they may manifest differently in different people. And if you sort of think back to where we were with the poly pill years ago, I think people were sort of hoping that there would be some aggregate benefit on lifespan or other endpoints, and it just never really got traction with anybody really to carry that forward. So I think you do need that first sort of stake in the ground, basically, to work around. At least that's from my perspective the most logical way of thinking about it. And that first one can be the most efficient one as well, so that you're not expending necessarily undue resources to get to a proof point that you can then leverage for other indications. Such is my thought. I think it's going to depend on the drug. It's going to depend on the use of the drug and the profile of the drug, but it's a way of going.
Yeah,
going after it.
Excellent point. That you have to get to an endpoint. You have to. We can't be there going forever on these big long-term endpoints, and we have to be able to knock off, show efficacy, and then develop the utility of these drugs even further. I mean, thinking about the right indication to go for first. Do you think this is very mechanism specific, that the difference of the aging hallmarks will be related to different indications first? And is there a sort of more sensible, low-hanging fruit indication to go for? And maybe I'll pose that to Joan. I know Joan and I have had discussions at meeting before where she thought I was crazy to go into Alzheimer's disease. And a lot of that was around thinking about the other biomarkers that we can collect as well. But Joan, do you have thoughts about what are the right first indications in this space that may help sort of open the door going bigger? You're on mute, Joan.
Sorry about that. I think it's really hard to develop drugs. It's really hard to cross the finish line, and you have to maximize your probability of success. So I'd say one of the first things to think about is, does the preclinical model translate to humans? And it's, if you can pick a disease where there's a higher chance of a preclinical model translating, your probability of success in the clinic will increase. And then second, you have to think, and Tom was getting to this, what does your drug actually do and where does it go? Because if your drug doesn't go to muscle, probably not a good idea to do a muscle disease. All these things have to sort of go into indication selection. And even if you're targeting, again, a pathway that might have benefit in all sorts of aging related, let's say, I spent a lot of time on TORC1, which I think is a great target. Your particular inhibitor of TORC1, as Tom was referring to, might not inhibit TORC1 in the brain because it doesn't cross the blood-brain barrier, or you need much higher doses to inhibit in the heart than you do in the kidney. So those things have to be taken into consideration. And so the dose of your TORC1 inhibitor for one indication may not be the same as your dose of TORC1 inhibitor for another indication. And so even within a trial, you may not be able to get efficacy signals in other organ systems. It's really complicated. So all of these things have to go into your thinking. And third, you do need to have a clinical trial path that isn't really long and expensive, that has an endpoint that the FDA will approve. And then what a lot of people don't think about, it's not enough just to get FDA approval. Payers actually have to pay for it, or no one will get your drug. And it's getting to the point where payers are saying, you have to show that you are saving us some money if you want us to pay for this. And that also has to go into the whole equation. So it's a complicated set of variables that have to be considered when picking an indication.
Drug development certainly isn't easy, and but we've got to go for it. We are going for it.
Absolutely, so but careful thinking.
Yeah. So David, you brought up going sort of more prodromal in disease states, which then ends up being very long studies, long clinical trials. How are you thinking about potentially breaking that down so that you can get to commercial reality early and perhaps build back? Have you?
Yeah. Again, the key here is to consider every opportunity on its own merits. And another way to think about what Tom and Joan have described is about the right target with the right drug for the right patient. And there's a good deal of resonance with what they've described. I think the hallmarks of aging are exciting. It codifies something for us, and I'd like to think that the hallmarks of cancer were published in 2000, I think it was, and our hallmarks in 2013. So maybe we're going to see a blossoming of activity, investment, and success, but it has to be through the right patient and efficient drug development. So we can't we have to use good translational principles, some of which Joan has described, and efficient programs. So, I would much prefer to being a program where I can prove that biology is important for a human being with a specific disease as early as possible, and then expand from there. Because what one finds is, of course, when it's the big step, right? The big attrition step is to prove novel biology is important in disease, and you can measure it by means of a clinical trial. And once you've got that, then I think investment tends to come in, and I think you can look to expand the waterfront on the back of positive clinical data. So I would always tend to go for the more efficient program, and that is one that expresses PK/PD in a meaningful disease endpoint as early as possible. And I think, what Tom described, I think he used the word stamps. And I think that's one thing we need to think of the biomarkers. But we need to take time and care to collect some of these things as we move along. Because also in a highly focused, capital efficient drug development program, you may not want to pay for a DNA clock. But I think there's something here about, if we're advancing the field together, we need to find a way to explore these things as we test the mechanisms in the clinic. And I think, ultimately, you've got to do things that are meaningful for patients. They move the dial. So good drugs tend to declare themselves early. We need to see that, and we need to be able to measure in order that a payer will fund what we're doing.
I think we're all very aligned in thinking there, and then the excitement of what we can learn on top from these trials, especially from some of the biomarkers that are being collected, is going to be an area of a lot of exploration, sort of learning new territories, but pretty exciting. So I'm still excited to hear, Joan, from the mTOR trials from Restore Bio. What are the biomarker changes? What are the depths there? So did you find that as a sort of an easy add-on to these trials, and something which is going to be weeding out and informing further in the near future? Is it a very tricky area to follow those additional endpoints?
You have to be careful to not overcomplicate later stage trials where you need to focus the study staff on the primary endpoint and making sure they are doing that correctly. So you don't want to add a lot of other complicated study procedures to your study. So we have to keep biomarker collection very simple so that it doesn't detract them from accurate collection of data. So I think that's kind of critical, is simple, like serum-based biomarkers or, RNA-seq kind of based biomarkers where you just have to collect blood and PAX gene tubes. A lot of FACS analysis and complicated procedures aren't deployable in large trials where we're probably going to need those big data sets to really understand aging biomarkers.
Yeah, I think it comes back to doing that rigorous drug development. You've got to keep your eye there and focus really well, and the side things are learning. I'm sure we'll come back to biomarkers, but there is a question that has come in about indication selection, and whether there are thoughts that AI will help improve indication selection, and the path forward, and maybe thinking about orphan indications that could come up. Tom, have you thought about the role of AI and indication selection?
Yeah, so actually it just happens that I've recently joined the board of a company that is sort of an AI platform that's looking for, basically they're leveraging their machine learning sort of capabilities to look for new oligonucleotide drug, basically landing sites of regulated protein expression. One of the things I think, first of all, I think the short answer is, in time, I think AI will really become a major force in a lot of the sort of, if you will, thinking about how to integrate complicated data sets and to provide sort of insights as to where they might be deployed. It's, I would say, in its very early stages, but where the data quality is high and where the data volume is very large, AI can play a very important role. And I think what sort of is the next major step from my perspective is the stitching together of electronic medical record and life experience, medical experience that's real out there with some of these lab-based measures or whole genome sequencing to really help to sort of steer people toward indications for which there are commonalities that can be linked to individual mechanisms. I think it'll come in time. I think we see some companies that are really focusing on developing that. It's big science, it's expensive, but my feeling is that the fruits will be there eventually. The quality of the data matter a lot. The quality of the inputs mean everything here. And so I think it's probably, many years off, but I do think so. I think there'd be a role where things are, I would say, where the light shines brightest right now for AI are in the areas where real information is fully digital and high quality. And so this, I think, is where DNA being a digital readout, where genetic variants being digital in their own sort of, by their very nature, this provides, the best link to what AI can do. And likewise, where you are manipulating the system through a digital drug, basically, which is how you could describe oligonucleotide-based therapies or RNA-based therapies. I think these are examples where I think things will come first, and then much later, maybe more complicated systems. But it's an area to watch. One of the things I've really been impressed with is just how few really highly skilled, highly trained machine learning scientists there are out there. They're in incredibly high demand right now, and they're being sort of taken away from biological sciences to work on things that are more, let's say, for some of us, pedestrian things like manufacturing and other types of processes. So I think having a focus on education of people in machine learning and AI for biological process endeavors in their own right is what's probably going to be limiting here. So I'll stop talking.
I just said that we're in a very motivating field, though, and I've had some very fruitful discussions there that people get excited about the prospects that we can get to these therapeutics, which are so broad in their utility, that the question is really motivating for a lot of people. And I'm going to add from our own experience, we're making progress using AI, particularly it's the high quality data, I agree, that matters. So we can tie down a lot in preclinical studies and actually lead ourselves to potential disease connections and allowing us to get informing orphan indications that we could go for. But then the quality of data is important as we go into clinical studies and start collecting plasma from those studies. You've got to keep the quality level high so that the information that comes out on the back end is really usable, faithfully usable to go into sort of following up that translational loop. So that's making sure, and as Joan said, you've got to focus on getting your endpoints right. If you want to really interpret from those samples using AI, going down that path, you've got to make sure you're going in with very high quality there. I think moving on a little from the sort of more traditional drug development paths, there are moves to be repurposing drugs and going into more nutraceuticals. Do you feel that we can learn a lot from these paths, which are maybe less traditional therapeutic approaches, or there are risks to going down that path? So David, maybe your thoughts on use of metformins, rapamycins, and then into nutraceuticals. Is this fruitful for us as a field?
Well, yes, although for commercial needs and for funding our research, it's always a challenge. I mean, I've been interested in repurposing ever since we repurposed Viagra in Pfizer for male erectile dysfunction to Ravatia for pulmonary artery hypertension. And we look pretty broadly, actually, because the PD5 enzyme was pretty ubiquitously expressed. But of course, some of the same challenges should not be overlooked, right? So with repurposing, biology is still often the major hurdle. You have to prove that the biology works in a new indication. But I think that the hurdle that's often not addressed is that there are two things really, I suppose. One is the company that wants to repurpose needs to determine what proprietary position they will bring to that opportunity. And this speaks to commercial realities, right? If I can't protect what I found and what I'm doing, then and the drug is generic, then it's going to be pretty difficult to protect it on the market in order that I can promise a return to my investors. And the second problem is if you don't own the drug and it's held under IP by another manufacturer, then of course you've got to gain access to it, and that can sometimes be problematic. So it's attractive if you can do it, because you can move very quickly into the clinic. And we've seen an attempt at that, obviously, through the COVID, the attempt to inhibit viral replication and change the immunological destination of patients through COVID-19. So I think that's kind of what I'd say on repurposing. I think in nutritionals, there's, frankly, kind of huge opportunity, really. We know if we're talking about longevity, I think it's in the nutritional space or the dietary space. We know that there are several interventions that are having an impact on health, well-being, and longevity. Just thinking about, I suppose, various fasting diets, the Mediterranean diet, and we're beginning to understand, I suppose, through an understanding of the science, what is it in these diets that is having that impact, and to an extent, even kind of the active constituents, even if not necessarily the mechanism. And a good example I've been thinking about recently are the polyamines, and like putrescine, cadaverine, and spermidine. All molecules that seem to be important for cell survival and growth, and you will know that these are molecules, Stephen, that decrease with age, right? And we also know that some of the foods in the Mediterranean diet and the Japanese diet are very rich in for example, fermented soybean, and which is rich in spermidine and other polyamines. And of course, then you can show that polyamines have an impact on fruit flies, worms, and yeast. And now we can look into humans. And we also know that they probably have an impact through impacting autophagy, which also declines with age. So, I think that's quite exciting. And the point really that I'm making there is, of course, that does allow us to move rather quickly, both preclinically and clinically, but also through nutritional supplementation and consumer measures to get, real measures in the clinic quite quickly. And I think we need to be quite smart about how we integrate kind of that nutritional space, what is emerging in terms of the science, and what we can measure quite easily, because patients, or because consumers, not patients then, they're consumers, are on these diets. And one can measure quite a lot now through wearables and simple capillary biomarker measures. So I think it's a terrifically exciting area that we ought to try and integrate into our thinking.
And just sort of following on a little bit from that, there's a good question from the audience here about can we improve the way and sort of accelerate the way that we move through clinical process to commercialization, potentially using AI simulation. So coming back to you, Tom, on the AI side, sorry, we're thinking on you on this side, do you really feel that we can also change the way we do this process through the clinic to commercialization through applying some AI principles there, as well as some alternative type approaches?
Well, I mean, the way I would put it is, I think the regulatory agencies are going to require real evidence that's driven in real time in populations that matter. For the sake of internal decision making within companies as to what they might choose to pursue or not, these sort of things could be useful. I think it's hard to know what sort of data you would need in order to gain confidence that what you're modeling is really going to be helpful in any decision-making context. But I think it's an interesting question to ask. I guess the AI infrastructure for that would need to be populated with large data sets. Just to give you a sense, before some of the things that that company I mentioned was working on were considered to be of sufficient robustness to actually become predictive, it took petabytes of data to sort of populate the models in order to drive that. So I think it's really going to come down to the fidelity of the data, the amount of data, and so if you have longitudinal data in humans that you can feed your model with and populate, so you can understand the arc of change of these different things with time, it might be useful. But I don't think it's ever going to help you in terms of getting regulatory approval. I think even for accelerated approval, it's unlikely to get you there. I think it's more likely that accelerated approval paths, which could be faster, would come from impact on very well-established markers of disease processes or of aging itself, or that are reflective of, as Joan put, the chronobiology of aging, or the chronobiology of these diseases. I think these are all things that could be done, but I don't know how the others feel about it. But my feeling is that we're a long way from where we are today with the medical practice and clinical trial design, to the point where we could say, do simulations that would be, of high enough fidelity to drive decision making at any approval stage.
Joan, maybe do you have other thoughts on accelerating the clinical process? I think you made very strong points about we've got to do rigorous drug discovery and drug development. That's an absolute necessity. But are there going to be evolutions in the way we can perform those clinical trials? I also would see AI as being something which complements, it parallels. It's not there yet to run a trial through virtual means or modeling type means, but it's something to help us improve the efficiencies. And is this a field that is amenable to sort of changing the thinking a little bit in clinical development so that we can be more effective?
Yeah, I am not an AI expert, but when I've talked to them, one of the messages I've got is you really need huge data sets to get meaningful, really robust conclusions from machine learning and AI. So I do think absolutely incredibly powerful, but what kind of data sets we'll need in order to mimic placebo or help clinical trials remains to be determined. And I think we'll need regulatory input and agreement on that. In a shorter term, I know Janet Woodcock has been discussing the problems with the multiple trials in COVID-19 that went nowhere and failed and couldn't recruit, and the importance of doing platform trials in that particular field. So having a protocol that's really robust and likely to get a meaningful endpoint in a certain aging-related diseases that we can use to test different interventions could probably speed up development in our space, because we won't then be reinventing the wheel and repeating each other's mistakes with trial design. And I think that would be an interesting approach.
I think, Stephen, if I may, I think we use, we've got to deploy AI sensibly. And I think that one can look at it as having to be absolutely correct. And the kind of the intimation in the question is that it'll replace a clinical trial. I don't think it'll ever replace a clinical trial. it certainly now allows us to model a clinical trial and its attributes, its endpoint, using Bayesian approaches. And of course, in PKPD, we've been using AI and modeling to predict where we will be with pharmacology for quite some time now. But in these other broader areas, I think within companies, we should look at it as a recommender system. So what it does is recommend something to look at. And in kind of drug discovery, that might be targets and pathways. But then you're never going to get away from the need of doing some smart wet experiments. But it allows you to target the wet experiments effectively. And as Tom was saying, actually, some of the wet experiments are really data rich, right? If you're doing single cell transcriptomics on a cell line that you're really interested in because of the indication that you're pursuing, you get a lot of data, and then being able to deploy that data back into a machine that can look at phenotypic and genotypic data in the patient subgroup, I think can be quite valuable. So I kind of think of this still as you've got a machine, and I can't remember if Joan or Tom said, I don't understand it either. It's a bit of a black box to me, but it makes recommendations. And then I know what to do with recommendations. I can test them. And I think the first thing it needs to do is to improve our probability of success as we move through the various stages. I think we're much better at testing pharmacology effectively than we were a decade ago, because of the use of modeling. And I think our clinical trials are getting smarter, both in measures of futility and responsivity of endpoints and selecting the right cohorts because of modeling. So I think it is making progress. I think we're a hell of a long way from the in silico human being and an approval. But I think we need to deploy it in the way I've described as a recommender system. I think often.
Great points, and just sort of feeding on. We generate a lot of very neat preclinical data, but we can also generate a lot of biomarker data in the clinic, and understanding the translational sort of relevance is very important as well, because a few-month-old mouse is very different from an 80-plus-year-old human, and understanding the translational differences is very important in the data equation here, what we bring in. I wonder if the panel have thoughts on the biomarkers that we have at the moment, and what is there a set of biomarkers which are really influential and worthwhile in understanding this translatability, and that can feed into some of these models as well to help us make decisions? Or is this an area which still needs a lot of work? And throwing more question on there, will we be in a world of a lot more companion biomarkers that help us move along a therapeutic to commercialization as well? So Tom, what do you think about the role of clinical biomarkers and where they will lead into the commercial sort of side of applying therapeutics?
Yeah, well, I would just say I'm a huge fan of it, and I think there are some really good and pretty comprehensive services out there that can provide hundreds of analytes of information with pretty high quality assays. For example, Myriad's platform I think is a good one for generating the sort of large range of markers. I mean, if you think about it, though, even 200 assays, it's less than 1% of the expressed genome, and so you're not looking at a lot of endpoints. Likewise, the lipidomics platforms that are out there and metabolomic platforms, all are very interesting, but I don't know yet where we are in terms of being able to use them. What I can say is that in my old company, which we're looking at a very interesting class of drugs that had anti-obesity effects, we sort of through that process stumbled on a couple of markers that were very, very robustly impacted by the drug. And these led us to basically studying the drug for impact on lifespan in mice, and it had a remarkable benefit there. Unfortunately, the drug was undevelopable for other reasons, but it led us basically to longevity and to understanding part of the mechanism by which the compounds acted overall. So I think, if the mind is prepared, having the substrate of the data to work on is really, you know, important. And so collecting it is useful. It is expensive. You do need to do it in a way that gives you the right type of sampling that teaches you something. I think you also need studies of long enough duration to see things happening. But I'm a huge fan of it. And you know, the thing that I'm always struck by is that the FDA must be sitting on a treasure trove of data if all of these things are submitted in the course of review of programs. And I would just hope that there is some way that they can use that to provide better guidance to companies as they're looking at these things. And, you know, it's almost like we need to look for these sort of veins of ore that are out there already that might be possible to be mined. So anyhow, I'll stop talking, but I think there's tremendous opportunity there and probably an overlooked opportunity generally.
Yeah, I think we all have great amounts of data, and being able to collaborate in a pre-competitive way in this would be very worthwhile, and making that happen is one of the things that I'm very keen on myself, that we can share data a lot more to be able to build this up. But then there is the question, are we all collecting biomarkers in the same way? Are we looking at data which is apples and oranges, really? So Joan, what do you think about the collaboration in this field, and how that will help us get to commercialization here?
No, I think you're right. I mean, the more data we can pool and then apply with those big data sets, the AI and machine learning, because you just can't process it otherwise, the better. I also think we have to remember the biomarkers have to be validated. So, you know, we don't want an epigenetic clock that with 3 samples from the same person gives 3 different ages. You know, we have to get really robust, meaningful, reproducible biomarkers so that we can learn something. And then I can just say, you know, even in our experience at RestorBio, it was the biomarkers that helped us understand what our drug was really doing. It sounds like maybe Tom's experience at Zafgen. There was a whole part of the picture that you couldn't tell from just a clinical readout, but you could tell from what was happening to the biomarkers, the pharmacodynamic effects of your drug that were very interesting and can help inform subsequent trials. So they are powerful, especially in a clinical setting.
Yeah. And David, I think to be provocative, the cholesterol of aging, are we going to have some equivalent like that in the future? Should that be something that we're trying to achieve, a biomarker that gives us that insight into a long-term efficacy? And you think that's sort of equivalent biomarker is coming in this field, or something?
Well, I think we need to look, because it would be liberating for the field and the science. But yeah, I think that comes really through a focus. I mean, as a drug developer, you need to focus on the biomarker that's most important to your target mechanism and disease. And in a sense, this loops back to the beginning of this conversation. What we do, I think, is to have the right focus, and a really good biomarker is important, right? Because, well, ultimately, it might become the cholesterol of cardiovascular disease, or the serum rhubarb of aging. However, having a biomarker that just shows that the drug is having the putative pharmacology, you can measure that it binds to the target in the right tissue, and there's some downstream effect, is so critical to a good development program. And there's data that's been published from well, my own group back in Pfizer, but also AZ, that shows that in the noughties, in the 2000s, of the failures in phase 2, we could only be sure we were testing the pharmacology in around 50%, because not enough attention had been paid to downstream pharmacodynamic markers, which at that point in time, of course, may or may not be related to the disease. But I think people it's almost like you want to run before you can walk. And actually, the first test of a good drug development program is, does the drug have a pharmacology? And that may not be on a biomarker that's specifically at that moment related to disease. That comes later through validation.
Oh, great insights. I think we're coming to the end of our time of this panel. So maybe just back to everybody for 1 minute of, we've covered a lot of territory, but we've really not gone deep into regulators, payers. We just touched the surface there. So maybe a final minute maximum from everybody, a big takeaway or a big thing that we should be thinking about for the future. So Joan, anything further you'd like to plan?
I'm just so excited to be in this space. I think, there's a lot of great minds and great targets that we're all pursuing. And I think we are really going to get some transformative therapies to patients following the path we're following. And, I think everybody on this panel, and I think most people in the field understand we need to be pragmatic in our approach. And I think we're going to get there and not in the distant future. So it's great.
Thanks so much. Tom, final thoughts?
Yeah, I'm with Joan. I mean, I would say also very, very excited to be in this space. I think for those of us who are in this area, it is so obvious that this is an important thing to do. It's achievable. It should be a moonshot that the whole world is really targeting, because it could have such impact on our civilization. And yet there's so many people who just don't get it. And so I think getting from where we are today with this sort of the skepticism or lack of interest or engagement that the really important people have, the key stakeholders, I think we need to kind of band together to drive that awareness. There's never been a war. There's been a war on cancer since the Nixon administration, as I always say. There has yet to be a war on these sort of major aging-related issues. And yet it's the one thing that we all face, every single one of us. And so I hope that we can get there.
And David, final thoughts?
Yeah, I mean, it's an extraordinary place to work. Where can you can span so much science and so much potential impact. And I think fundamental here is unhealthy aging, right? We talked about it at the start. And frankly, that's a public health crisis. I think Tom was making that position, position, and frankly, could overwhelm financially or in other ways the health services. And I'm optimistic because I see what's happened in the last year in the response of our industry and science to the COVID pandemic, and the fact that completely new platforms. I got injected with a new platform of vaccine that was 9 months in development. I mean, if you told me that this time last year, I'd have laughed at you. But the RNA vaccines are there and they're making a difference. My goodness, they're making an enormous difference. So we have a huge opportunity, and it's exciting to be with the panel and with great brains thinking about having that impact for people.
Great. So thanks so much to David, Tom, Joan. I think it's been a fantastic discussion. This is a field which is going to be very productive, and it's fun to work together as a team in this when you've got such a big mission and we can really team together on this. So I think we all look forward. We appreciate the Angela and the longevity leaders meeting, bringing us all together and helping sort of facilitate these discussions really well. Keep us having fun and keep us getting to that endpoint as well. So I'll close this panel right now and pass back to Angela. Thank you very much.
Thank you, Stephen. And look, thank you for the wonderful work you've done moderating today's discussion. Of course, thanks to the panel. I'm not sure about our audience, but I feel really excited and energized after listening to that discussion. It's wonderful to hear so many great brains working in this space, thinking clearly and really bringing a new form of medicine to the world. So looking forward to following how this unfolds. I hope you'll stick around with us for the final closing session of our Aging Science stream. We have a real treat lined up for you, looking at creating consensus on a biology of aging paradigm with some more very, very prominent thought leaders in the space. So do stick around for that. It's worth noting for any of you who have missed some of the sessions over the last couple of days, this is our last day of content, live content, but everything we've done has been recorded and it's all available, staying still on the platform. It'll still be there for you to revisit up until next Friday, the 14th of May. So please feel free to revisit anything that you have. The partnering system is still open, so you can still connect with one another as well, again, right up until next Friday. So it's a slightly different way about how we consume content in the virtual world and make partnerships in the virtual world, but we hope that it's still enabling you guys to do that. And fingers crossed, we see you all together live next year. But without further ado, look, thanks again for watching. Thanks for being so interactive to our audience. And thank you again to Stephen and our panel for the wonderful insight. Bye-bye for now.
Bye. Thanks, Stephen.
Thank
Bye.
you.
Thank you.
That was great.
Thank you.